Hello friends, today we were discussing about the isoenzyme. Isoenzyme means the multiple form of enzyme catalyzing the same biocatalyzing reactions but different in the physical, chemical, structural or immunological properties pH, Km and vitamins. These have a difference in properties but they have a same the biocatalyzing reaction. It is called as the isoenzymes. For understanding these isoenzyme, let us to take a one common example. When you visit to sweet shop, that is bakery, in that bakery so many varieties of sweets are available. All the varieties of sweets give a sweeten in taste but they have a different in names. Here the different name is have refers to physical, chemical and structural, and but the same sweeten in taste. All varieties of sweets they give a same taste, that is the same biocatalyzing reaction. Multiple form of enzymes they give a same biocatalyzing reaction but they have a different in a physical, chemical, structural, etc. It is called as the isoenzymes. And the properties of the isoenzyme, the characteristics of the isoenzyme synthesized from different genes. Examples: the malate dehydrogenate, it's present in the cytosol, that is cytosome and mitochondria. Some isosomes are the oligomeric. Oligomeric means it's made up of a more than two subunits, and some enzymes, isoenzymes are monomeric. These are made up of only one subunit. It is called as oligomeric enzymes consisting of a more than one type of subunits. The example is lactate dehydrogenase and creatine phosphokinase. These two are examples of the isoenzymes. An enzyme may be active as a monomer or oligomers. The enzymes when the active in the form of a monomers or oligomeric. Example is glutamate dehydrogenases. And here the first example of the isoenzyme, that is lactate dehydrogenase, that is we call as LDH. LDH, whose systematic name is L-lactate NAD plus oxidoreductase, whose enzyme code is 1.1.1.27, catalyzing the interconversions of a lactate and pyruvates. It is the structures of the lactic acids and which interconverts the pyruvic acids in the presence of an enzyme, that is it is a isoenzyme LDH. The few types of isoenzyme of LDH. The LDH, it is an enzyme but it's having the different property. Here the five types of isoenzymes of LDH. The properties of the LDH is the tissue isolations. The example, that is LDH, its properties tissue isolations, and they have a five types of isoenzymes: LDH first, LDH second, and LL and LDH5. And the LDH first is the most positive charge and fastest in the electrophoric mobility, while LDH5 is slowest, and they can separated by a electrophoresis. Here these LDH, they can tissue isolations of tissue, they can separated by using the one method, that is electrophoresis method. With the help of this method we can isolate the tissues. And these five classifications of this isoenzyme of LDH. LDH first, the subunit is H4 the originated from the heart and RBCs, and the second one LDH2 and the subunit is H3M originated from heart and RBCs. Third iso enzyme of LDH, that is L, and the subunit is H2M2 originated from brain and kidney, and fourth one is also L and subunit is H and M3 originated from the skeletal muscles and livers, and the last H5 the subunit is M4 and it is originated from skeletal muscles and livers. It is the first examples of this isoenzyme, that is lactate dehydrogenase. The second example of the isoenzyme, that is isoenzyme of creatine phosphokinase. It is the second examples of this isoenzyme. In this topic of isoenzyme, the examples are most important. In the exam point of view, the question may ask like, read the define isoenzyme and explain the examples of the isoenzyme. At that time, you explain the first example, that is lactate dehydrogenase, and second example, that is creatine phosphokinase. The creatine phosphokinase, or we call as creatine kinase, that catalyze the interconversions of phosphocreatine to the creatine. The phosphocreatine is converted to creatine in the presence of the enzyme isoenzyme, that is creatine phosphokinase. And here the structure, that is CPK, is nothing but a creatinine phosphokinase are exit in three isoenzymes. First isoenzyme, that is CPX1, that is subunit is brain and brain, the originated from brain. CPX2, the subunit is muscle brain, originated from heart, and the third one isoenzyme of subunit CPX3, the subunit is muscles and muscles, originated from the skeletal muscles. The second one, that is therapeutical applications or uses. Streptokinase or urokinase causes the lysis of intravascular clots. These two isoenzymes are causes the lysis, the breakdown of the intervascular clots, and it is used for the treatment of the myocardial infection. It is the first uses applications of. Second one, aspergillus is used as an anti-cancer drug used in the treatment of leukemia. And third one, pepsin and trypsin are useful in the treatment of chronic indigestion and the pancreatic insufficiency. And the fourth one, that is collagenase enzyme, is used in the degradation of the collagenous tissue and used in the severe burns and ulcers. Last one uses, that is lysoenzymes, is found in a human tears. In our tears, the lysoenzyme is present. It acts as an antibacterial property and used in the treatment of eyes. These are the therapeutical applications, and in the next class we are discussing about the coenzyme. Thank you so much guys. I hope you guys understood this video. Don't forget to like, subscribe